a short version of it, and then we'll go into the Q&A part of it. Okay.
Okay.
And you can go ahead and start.
Okay. Hello, everyone. Thanks for attending another one of Universal Sequencing's Q&A sessions. As Aaron mentioned and alluded to, we've had a pretty big uptick in people new to Universal Sequencing's technology show interest in this forum. So I'm going to take the first 15 or 20 minutes to give a very quick overview of the technology, as well as a handful of representative applications. Hopefully this will be a good learning experience and or refresher. If you feel like you are fairly familiar with universal sequencing as a technology, now would be a good time to get a coffee, because I'll be just going through some slides pretty quickly over the next 10 to 15 minutes. So I'm Andrew Anfora. I am the Senior Field Application Scientist Manager. I'll be doing the bulk of the presentation. We have Aaron Levy, who's our Chief Commercial Officer, also present. David Nordell is our Inside Sales Manager. He's dialed in. Certainly not last but not least, Tom Chen is our Chief Scientific Officer. Didn't have his name listed on this particular slide, but he will be here for answering questions as well. So I'm just going to launch right into it. Feel free to type questions into the chat as you think of them. We're going to go through the presentation first, and then we'll address questions. David will be monitoring the chat. David, if you see like a lot of questions on a given slide, feel free to interrupt me. Otherwise, I'll just blow through this.
Will do.
All right. So just to summarize what we're going to be talking about, Universal Sequencing produces TELSeq, WGS Library Prep Kit. This is a simple, fast, one-tube library prep assay for long-read sequencing. It converts short-read sequencer data into highly accurate long-read sequencer data. And we can capture molecules greater than 100 kb in length. Relative to most library preps out there, especially in the long read space, we are a very low input assay, requiring sub-nanogram to nanogram quantities of DNA. And I'm going to explain the biochemistry of our library prep, as well as cover some applications, including de novo genome assembly, a preview of our metagenomics analysis pipeline, go over some examples of structural variation, whole genome phasing, and example of targeted phasing of exomes. So to hit on our technology, this diagram should largely be at least 2/3 review for everybody in attendance. What we have is a representation of different flavors of next generation sequencing, where we have a diploid organism represented with one chromosome colored blue, the other chromosome colored red. With short read sequencing, you get highly accurate short reads of 100 to 500 base pair that are also relatively low cost to generate. The challenge with unlinked short reads is it is nearly impossible to phase haplotypes, certainly if they are further than 500 base pairs apart. It's really difficult to confidently say that two given variants are present in the same chromosome. Additionally, de novo assemblies are quite limited and can't achieve contigs of significant length if you're only using short reads. One way around this is to use long read sequencers such as PacBio and Oxford Nanopore. These sequencers can generate more longer read lengths depending on the instrument and the run mode, looking at highly accurate reads of 10 to 15 kb. In the case of PacBio, you do have the option of running in a lower accuracy mode to get slightly longer length reads. And the tens of KB read lengths do allow for some degree of phasing of a genome and a reasonably robust de novo assembly. A linked read solution such as TELSeq is taking short reads, but prior to fragmentation will be applying a UMI to the intact long molecules such that the library fragments can be traced back to a parent molecule, and we can therefore link the reads, preserving a lot of that large molecule information with the high base calling accuracy of a short read sequencer. And we are able to capture molecule information upwards of 100 KB, and I'll have a couple of bits of data later on supporting that claim. I'm going to go very quickly over our mechanism of action. I don't want to spend too much time on this, but here is how we actually generate our libraries. Really an important thing with our process is we don't have any exotic capital equipment associated with our workflow. Everything takes place in PCR tubes and a magnetic stand. If you're already running NGS library preps, you won't need any exotic equipment to start using TELSeq. What we do is working from left to right on this diagram. We start with mixing a transpososome complex and our TEL beads. The TEL beads each contain a unique 18 base pair barcode. So we have a potential barcode library of roughly 2 billion sequences. So we mix the beads and the transpososomes, and then we add high molecular weight DNA. Ideally, we want to be loading DNA molecules 40 KB and above. You can still get some usable data out of TELSeq with molecules as small as 10 KB. So depending on how you are preparing your DNA samples may already be fine, or you may need to do some minor adjustments to isolation to really maximize the data coming out of a TELSeq assay. So once the beads, transpososomes, and DNA are mixed, they will form a complex to which we add a second transpososome for tagmentation. We will break and wash the DNA, and then we generate amplicons using sequences on the two transpososomes. So the subset of your long DNA fragments that contain the proper orientation of the transpososomes will be amplified into Illumina compatible libraries containing your P5 and P7 tags. This is a very fast workflow. From isolated DNA to library ready for sequencing takes only 3 hours. So it would be possible to go from biomass isolation to initiation of collecting data all within a single workday. I'm not going to spend a lot of time on this. There's actually a fair bit of detail here. I do want to mention that we provide analysis software for free, either as downloadable Docker or Singularity images, and some of our tools are currently available on BaseSpace, and we are going to continue to be adding our software tools to BaseSpace for those that prefer doing their analysis on BaseSpace. So all of our universal sequencing provided software is depicted in green on this chart. Our data type are very similar to 10x Genomics legacy linked read offering. So we do also provide a conversion tool that converts our 18 base pair indexed format into a 16 base pair indexed format that is compatible with 10x Genomics tools. So if you're used to using Long Ranger or Supernova for linked reads, you can continue to do that. And not shown on this diagram, after running our QC tool TELRead, you can take those FASTQ files and do some third option not shown. So again, just to summarize the functionality of the assay, it is a 3-hour library prep using very low DNA input. So sub-nanogram quantities for microbial and targeted panels, and up to 5 nanograms for larger genomes. We're able to capture super long fragments and preserve size information for molecules greater than 100 kb in length. So really, even amongst assays that are doing some type of read linkage, we are capturing much longer molecules than pretty much anything out there. And our software is free. Just for comparison sakes of the DNA input, just a reminder, so when we're talking about 0.1 to 5 nanograms of input, if you're using a actual long read sequencer, you'll be needing microgram quantities of DNA. So we've had some users be able to enjoy using TelSeq because they're able to do DNA isolations from a single insect and still have enough DNA to create that single organism de novo genome assembly. So now I want to quickly go through some data examples before opening things up to questions. I'm not going to spend a lot of time on this because it's available online. We have collaborated with Illumina on several fronts, and one collaboration was in microbial de novo genome assembly. And this is an application note available on the Illumina website where it was describing the utility of TelSeq as well as the availability of some of our software tools on BaseSpace. Very briefly, we looked at a wide range of bacterial genomes, including high and low GC organisms. So we are able to handle high GC organisms. What you can see on the chart to the right is higher GC organisms will have slightly lower library yields. Just we are using a PCR step to amplify our libraries. So as you'd expect, slightly less yield with the very high GCs. I also like this study because we were DNA fragment sizes of 20 kilobases were used. So that's actually smaller than optimal, but they were still able to get really great data. This is a very busy chart. I'll just say the top part is describing the genomes that were used, so it's the reference genome information. The middle part of the chart is looking at the actual data collected. Illumina was collecting 100x coverage. I believe that is what they need to do if you're assembling a unlinked genome. For our workflow, we typically recommend starting at 50 to 60x coverage, and that's often sufficient for a high quality assembly. This data set had, as I mentioned, molecules of approximately 20 kb in length in populating the data sets. When we say SLF there in the middle two rows, that's an abbreviation for super long fragment, and that is the computed fragment length calculated by our software. And in the case of these microbial genomes, we were able to assemble the majority of the genome, the majority of the chromosomes into single contigs. We were also able to assemble the majority of plasmids for the organisms that contained plasmids, and we're able to produce data sets covering typically 99% of or greater of the genome. The worst we did with in this study was 97.28%. So we're really able to assemble near as no difference the entire bacterial genome. So yeah, single contigs. We were able to de novo assemble plasmids. So if you have total DNA isolation containing plasmid, you'll be able to assemble the plasmids into contigs as well. And just to briefly contrast what a microbial genome assembly looks like with TELSeq compared to a Oxford Nanopore dataset and unlinked data with Illumina. You can see that there is that tremendous difference in DNA input, just night and day, less than a nanogram versus a microgram for the other options. We are assembling the genome into a single contig, so largest alignment and largest NA50, of course. Since we're using the Illumina base calling, we have no misassemblies, very few mismatches per 100 kb, and very few artificial indel artifacts in the data set. So really a quite robust single microbial genome solution. So we are so good at single microbial genomes, we have started to look at doing metagenomic samples. This is a tool that will be forthcoming. So very briefly, if you have a microbial community sample, you would follow the same initial steps of our workflow. So we'd still prep the library, you'd still run our QC tool, and you'd still run our de novo assembly tool. We are bringing online a third software tool for this workflow that will assign strain level identification and quantify abundance. So we've been testing this with some of the ATCC microbial community mixes. In this case, it's a summary of a 20 species staggered mix, where represents a community with pretty high dynamic range of species abundance. So far, we have been able to handle the 3 most abundant groups of microbes quite well, able to assemble and assign identity to all of the more abundant species. The species that are present at a fraction of a percent, we're able to identify their presence, but coverage becomes limiting for the de novo assembly. So we're still building out our guidance for recommended dynamic range and really what best practices would be and what you could reasonably expect for your microbial community sample. So we're very close to having a tech note out, and we have been presenting a poster with some of these data at a few conferences. Going to move quickly on to phasing of human genomes. Really quickly, just going to say that most of our human genome work is in 12878 and 24385, the genome in a bottle samples, as you would expect. Our data sets typically have the overwhelming majority of molecules being greater than 20 kb in length, with a significant portion of the molecules being greater than 100 kb in length, and that really lets us do some of these robust phasing and structural variant calling analyses. We have really good recall and precision rate for both SNPs and indels. As you'd expect, a little bit harder to call indels than SNPs, just like any other platform. Not going to spend too much time on these figures, but these data are from our genome research paper, which is really a great comprehensive description of what TELSeq can accomplish. Just briefly, we were able to phase the MHC region, and here is a depiction of the variant assignment for some of the class 1 HLA and class 2 HLA genes. The regions where you can see a miscoloration or what appears to be an incorrect assignment are in regions of lower confidence in those references. So these are tricky genes to phase, and likely some of these assignments of being called class is incorrect are likely as much a reflection of the reference as it is this data set. Additionally, just another example of why it's important to phase, what you can see. Here's another region, and this is also in our genome research paper, where we have a two deletions of different size in the different chromosomes in the same region. So we had one 19 kb deletion in the first haplotype, and the second haplotype has a 114 kb deletion. You could imagine if you weren't able to robustly resolve the haplotypes, you would probably only be able to detect that 19 kb deletion and just think you had some low coverage for the remaining 100 kb of that 114 kb deletion. So we can phase diploid genomes well, even in the tricky regions. Just wanted to briefly show the relationship between DNA molecule size and observed phase block. Similar trend would hold true for our de novo assembly applications. So what you can see here is the exponential gain in phase block size relative to input molecule length. So it's really been data observations such as these that lead us to the recommendation of using DNA input molecules, ideally 40 kb in length or greater. Depending on the specific project need, you still may be able to get a contiguous enough data set for your need with molecules shorter than 40 kb. Generally, we recommend that you use at least 10 kb DNA fragments into the library prep. We'd kind of expect some pretty diminishing returns after that. So again, even if you can't get 40 kb DNA molecules, you still may see some benefit from TELSeq. Molecules greater than 40 kb are going to give you the best resultant data. So just to go over a couple examples of scaffolding really quick. So in the de novo assembly space, a lot of users already have pretty good PacBio data sets or pretty good Oxford Nanopore data sets. We've found that our ability to capture those 100 kb molecules, as well as having a different biochemistry of library preparation and having different breakpoints in our assembly, there's still really good value in using TELSeq to further scaffold an existing genome assembly. So here's an example. Here's two examples of, again, a pretty good PacBio assembly, a pretty good Oxford Nanopore assembly that was made more contiguous by adding TELSeq data to the assembly. And of course, you could also just use TELSeq for a de novo assembly as well. I'm leaving out examples of that just for the sake of time. Yeah, I already said that. I just want to close with an example that we are also able to phase exomes. So we have a protocol for phasing or for using TELSeq with SureSelect. Essentially, you run our TELSeq protocol with some extra PCR cycles to ensure there's enough product. And then you go right into the Agilent SureSelect workflow. The only modification needed is using blockers that we provide rather than the standard SureSelect blockers. And we're then able to phase the majority of the exome. And again, we have a protocol for using TELSeq with SureSelect that's available on our website. Yeah, so we're able to phase exomes. We've also done some other enrichment technologies that I'm not showing for the sake of time, and just want to close by saying that we have offices in California and Massachusetts, but we are supporting the world. We have customers all over the globe. I will pause there. And David, let me know if what questions I should address with Tom.
All right, thank you, Andrew. So let's see here. Again, if you guys have any questions, please feel free to type them in chat. We do have some questions that were kind of pre-submitted. So let's go to the first one. How many samples can be run with one TelSeq kit?
So the number of samples per kit is going to be a little bit dependent on the genome size. So we have our standard kit is sufficient for 12 microbial genomes, 4 human-sized genomes, or an intermediate number of intermediately sized genomes. Our protocols bin the genome sizes into 4 distinct buckets to advise how much reagent to use, how many beads to use. And then we also do have a high throughput kit that is 6 times the volume, so enough for 24 human genomes or 72 microbial genomes.
Thank you. Let's see, second one we have. You mentioned the smallest DNA molecule size that works is 10 kb. What happens if you try to sequence small genomes, such as those as eukaryotic viruses, 2 to 5 kb, or metagenomes of varying sizes, 2 kb to 1 MB in length? That's the first part of the question, and I'll let you answer that, and there is a second part.
Okay, I'll start, and then Tom, I'll probably, if you're on, you can fill in if there's any more details. There's probably two major challenges with using shorter molecules. Actually, the major challenge with using shorter molecules is we don't want to be overloading the beads. So one thing that will happen is if you have as you get shorter and shorter molecules, you have obviously more discrete DNA molecules per unit mass of DNA in your sample. It is possible to overload our beads if we put too much DNA, too many DNA molecules in the sample. You will actually have fewer contacts per molecule with those UMI barcodes. So you can load a lot of DNA onto the beads, but then you're going to be decreasing your signal per molecule and will effectively be artificially shortening your assemblies just by not collecting as much information per molecule. And Tom probably has more to add.
Yeah, I think Andrew made a good point. So that's why the molecule to barcode ratio. Another reason we emphasize so much, we want the 40 kb or even 100 kb input. That assumption is we talk about we sequence the complex genome is so big. It's a megabase, 100 megabase. However, if your genome is only 40 kb, yeah, or even 10 kb, we definitely don't need that long. So it depends. For some people, only interested in some very small genome. The size pretty small. It's even under 10 kb or less. So that's for TelSeq, it's okay. TelSeq can work efficiently for the fragment several kb long. But keep in mind, with that shorter fragment, the capability to assemble much longer will be much less. So it's all depending on your application. If your target itself is only under 10 kb, yeah, it's several kb fragments long enough to assemble those back. But if your target fragment is a megabase, billion base, yeah, if you only use 1 kb fragment to assemble that will be challenged. So let's go back. The issue is now if you're mixing both long one and short one together, from chemistry point of view, it will be really hard to really efficiently capture those long ones. So that's make all chemistry, the barcoding tend to more efficient to capture more and more the short one. So that's why overall, if in your mixture, you indeed target for the large genome, you want to get the best assembly, best facing in those sample. If you have a lot of 1 kb, even 3 kb, those shorter genomic DNA we call it contamination over there. You can still sequencing. However, you're going to be imagine you're going to waste a lot of reads, which very short or very short linked molecule will not be very efficiently to facing the large region you're looking for. Yeah, so moving forward, we actually now we developed some specific application for the long range PCR template. So that's usually under 10 kb. For those ones, we need to pay attention to make sure we control the molecule number to the beads to control the loading. The second is we can scale down the input, also reduce the reaction cost. By doing that, we can get a pretty good the facing on those short, relatively short DNA. As far as several kb considered short, but for standard the sequencing several kb already pretty long.
Thank you, Tom and Andrew. Okay, so next, and that kind of leads into this next question, can I mix multiple libraries together for sequencing? For instance, if you have 3 fish species that you're trying to, 3 libraries from fish species that you're trying to sequence.
So I'll start, and then Tom, you can throw out some additional caveats if I miss them. So with our workflow, and actually I'll jump to a picture of the libraries. With our library structure, you have, so our index 1 is the 18 base pair bead specific UMI. Then our index 2 is an 8 base pair multiplex primer that's going to be library specific. So from a functional standpoint, you can we sell 24 different index 2 oligos. So you can multiplex up to 24 samples on a given flow cell if you're getting enough throughput off of the flow cell. Obviously, the best case scenario is to be sequencing genomes of similar size with TELSeq on the same flow cell. Tom, I don't know if you want to mention any of the specific concerns if we're doing mixed runs of different size organisms.
Yeah, I think different size organism is not much different because for sequencer, it doesn't care about what's your genome size as long as your library look like a similar structure. Right? It's indeed a tricky part. If the genome size, just like Andrew mentioned, vary too much, then you have to make sure you place the right ratio, because the smaller genome for sure you only need a few reads. Large genome you need a lot of reads. So that will make the mixing a little bit tricky to get the right ratio. Other than that, yeah, I don't see any reason why you cannot. Actually, most people do the pooling because these days NovaSeq is just too much, too powerful. Yeah, there's no way you just want to use one NovaSeq to one sample.
And just I'll quickly add, our libraries do require a spike-in sequencing primer, but that primer is compatible with Illumina libraries. So it's also possible to multiplex a flow cell with both TELSeq samples as well as standard Illumina libraries.
Thank you. Okay, and that kind of again leads into this next question. Can I use TELSeq data with other long read pipelines?
Tom, I'll let you take that one because you've seen more scenarios.
Yeah, it depends on what that means. If you mean use both data type for the analysis. Yeah, well, one the example I think Andrew kind of alluded to. People we can use other long read to make an initial contig. That you can use the TELSeq to do the further scaffolding, or you can use TELSeq because TELSeq again remember use Illumina short read have high accuracy. You can also use TELSeq read to do some gap filling. It's for sure that's a different way to play with it. To truly get perfect genome, you do need a multiple platform to take advantage of what we can offer. They can offer. Yeah.
Thank you. And how is TELSeq linked read different from a normal long read?
So
Andrew, do you have a slide?
yeah, I'll go back to all right. So really, what we have depicted here is the difference between what the reads look like at a sort of bird's eye view cartoon level. So if you look at the middle of this chart, there's a representation of long reads where you're getting, let's just say 10 KB reads. What we're getting with TelSeq is those your Illumina libraries. So we're typically looking at a 200 base pair library fragment with our default workflow. But what we're doing is prior to fragmentation of the long DNA molecules, that is where we are labeling the libraries with the bead-specific UMI, such that we know when we see the short reads, if they share the same UMI and are mapped to within 50 KB of their nearest neighbor, we know that they come from the same molecule of origin. So we're able to say with high confidence that any number of short reads sharing UMIs within 50 base KB of their nearest neighbor are going to represent the same molecule. And so we are able to informatically reconstruct these longer molecules, even though we're using shorter fragments.
And not depicted in the diagram here, but a similar follow-up would be how does that compare to a synthetic read?
So we are a quote-unquote synthetic long read platform. There are several out there. I would say at a high level, at a very high level, we're functionally similar to many synthetic long reads in that we're preserving long molecule information prior to fragmentation. I would say the major advantages of TelSeq are severalfold. We are a very fast workflow that uses low input, and also our protocols are optimized to preserve very long molecules. So if you look at several other, very typically for linked read products, they're often targeting that 10 to 20 KB fragment length, trying to approximate a PacBio read using an Illumina sequencer. We collect those data, but we also have a high percentage of molecules substantially larger than that. So really getting from those human data sets I was showing, we had 20 to 30% of the molecules greater than 100 KB in length. So we're really going beyond the read length that you'd get from a PacBio sequencer, for example, and also where the majority of your molecules for Oxford Nanopore aren't going to be that long either. And also other synthetic long read platforms really tend to gravitate more towards the 10 KB space.
Yeah, let me add one point is, Andrew mentioned the synthetic long read in a broader definition. There's another way definition for synthetic long read, much narrow, which refer to the way how they really use this short read. The purpose is they actually at the per molecule base, after barcoding, they want to reassemble back to the original molecule. So those ones, narrow speaking, is a synthetic long read refer to they have the intention and the capability after barcoding, then go back to reassemble the original per molecule to get full length coverage. By doing that, limited the power to get the long range information. That's the reason is most standard synthetic long read only can get around 10 KB range. For us, we have the advantage. Our TelSeq is a more standard definition as a link read, has the power to get much longer range the sequencing information over even 100 KB. But the drawback is exactly like the drawing what we show you here. You actually see the bottom one, the short read what we having, yeah, they have a long span, but they do have bigger gap in between. So per molecule level, we are not able to assemble back to the 100% coverage. We really relied on another link read molecule to overlap. So eventually, the whole scheme, yeah, we can get the full coverage. But per molecule level, we don't have intention to assemble back our small read directly to the original molecule. So this probably fundamental definition is a broad-based synthetic long read and a narrow-based synthetic long read. Personally, yeah, I would like to just use narrow definition synthetic long read to differentiate what we have as a link read.
Thank you, Tom and Andrew. Okay, I think this will probably be the last question we have for you. What is the best way to prepare the DNA for the TELSeq kit?
So that's going to be a somewhat organism specific. We have some guidance for certain sample types. Generally, you want clean DNA as long as you are able to isolate. So there are several kits. There are also several homebrew gentle lysis methods. I guess if there's a specific organism of interest, probably email our tech support email address, and I can get back to you with more specific guidance. There's an increasing number of kits that are able to isolate sufficiently high molecular weight DNA. Generally, any kit that says it can isolate high molecular weight DNA is probably going to give usable DNA. There isn't a hard definition of high molecular weight DNA with regards to how vendors will market their kits. So there may be some that are going to get you longer molecules than others, but it tends to get organism specific.
All right, thank you both again, and I think that's it for the questions we have.
Well, in that case, we can wrap this up for today. Thank you for your participation, and we will see you next week.
Great, yeah.